Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. Today in Embryology Foundation module lecture 8 we are going to discuss with the first week of development. So first week of development, the cleavage is the first major phase of embryonic development. It consists of repeated mitotic divisions of the zygote, resulting in increase in number of the cells, now called the blastomeres. So there is a rapid succession of the cell division, and it creates a multicellular embryo from the zygote. It partitions the multicellular embryo into the developmental regions. So cleavage, by definition, the series of the mitotic cell division of the zygote to form embryonic cells called the blastomeres. And implantation is the process during which the blastocyst attach itself to the endometrium. So in this diagram you can see this is a 2-cell stage embryo which is formed after the fertilization, and the first cleavage takes place and becomes a 4-cell stage mass, and then after another cleavage division, another mitotic division leads to 8-cell stage. Then at the third mitotic divisions, 12-cell to 16-cell stage, the cell to 12 to 16 cell stage the embryo is formed. So you can see through this diagram that with each successive division, size of the cell is reduced and number of the cells is increased. Also at this stage, after the third cleavage division, the cells maximizes their connection with each other via gap junctions, and the process is called the compaction. And at this stage it is called the morula. And after this morula stage, it enters into the uterine cavity, and small amount of the fluid enters into the cavity, and then when this fluid forms a cavity inside the morula, it forms a blastocystic cavity, and it divides then the substance of the embryo into an inner cell mass and an outer cell mass, and the structure is called the blastula or blastocyst. So in this diagram you can see this is the blastocystic cavity or blastocele. This is the inner cell mass which is called embryoblast, and this inner cell mass will form the embryo proper, and this area is the outer cell mass which is also called the trophoblast, and this outer cell mass or trophoblast will form the extra embryonic membranes and the placenta. So in this diagram you can see the time of the ovulation. There is the release of the oocyte. Here the fertilization is taken place and zygote is formed. Here you can see the 2-cell stage, the 4-cell stage, the 8-cell stage, and then the morula. This morula resembles the raspberry, that's why given the name. Here you can also see still at this stage the zona pellucida is present, but as it enters into the uterine cavity and fluid enter into the intercellular spaces of inner cell mass, the size of the morula increases, cavity forms, it becomes the blastocyst, and at this stage also the zona pellucida disintegrate, and the process is called the hatching. And after this hatching process, this blastocyst again reorient itself in such a way that its embryonic pole where the embryoblast is present, it faces the uterine cavity and it starts embedding, and the implantation take place. So in this diagram again you can see the first cleavage division, the second cleavage division, the 4-cell stage, then the early morula, the advanced morula, and the blastocele which has now started to process of the implantation. So this is a first cleavage forms the 2-cell stage. It is 30 hours after the fertilization. This is again your MCQ as well. Then they split into form the 4-cell stage embryo. After the 4-cell stage they form the 8-cell stage embryo, and then eventually morula forms. And after this morula, the small amount of fluid enters into the inner cell mass where it forms the blastocele, and the embryo is now given the name of either the blastula or the blastocyst. So cell division after the fertilization, the diploid zygote undergoes a period of rapid mitotic divisions called the cleavage. Here you can see all these mitotic divisions. These are controlled by the cyclins and the cyclin dependent kinases. During cleavage, the zygote is divided into smaller and smaller cells called the blastomeres. So marula is about 3 days after fertilization. This picture shows you marula, a solid ball of 32 cells that resemble the raspberry. In this diagram you can see the zygote, the early cleavage, the marula, the early blastocyst and the late blastocyst. In late blastocyst you can also differentiate between the inner cell mass and the outer cell mass. So blastocyst stage, two distinct types of the cells are present. The inner cell mass forming the embryo. Here you can see inner cell mass which is forming the embryo, and the trophoblast layer are the outer cell mass, the layer of the cells surrounding the cavity which help form the placenta. Here these are the trophoblastic cells which surrounds the blastocystic cavity. At this stage the blastocyst it floats into the uterine cavity for the 3 days. So implantation usually starts 6 days post conception. The trophoblast erodes the uterine wall and it takes 1 week to complete. If inner cell mass of the single blastocyst divide, the monozygotic twin also form. This is one of the type of the twinning as well at this stage. Another point you must remember at this stage, as the implantation takes place 6 days post conception and it takes almost 1 week to complete. So there is a little amount of the bleeding at this time which is called the implantation bleed. Sometimes females sometimes misunderstand this implantation bleed as their menstrual cycle and their dating then affect. So they do not understand, they do not know that the pregnancy, the fertilization has taken place. So in this diagram you can see the inner cell mass, the blastocele, blastocystic cavity and the trophoblast, and it has started to invade over here. Inner cell mass forming embryoblast forms the embryo proper, and outer cell mass, the trophoblast, form extraembryonic tissue and primarily the placenta. So blastocyst, in this stage you can see the blastocyst, it helps attach the embryo and helps in implantation of the embryo. Here you can see the inner cell mass and the outer cell mass. Outer cell mass also differentiate itself into 2 layers. The outermost is the syncytial trophoblast and inner more cellular layer is the cytotrophoblast. Coincident with the blastulation, the zona pellucida is shed. The process is called hatching. Hatching exposes bare trophoblastic cells to the uterine wall, and uterine wall is prepared for implantation each month under the hormonal control. So the trophoblasts begin to thicken as it begins to implant, and the L-selectins are the present on the trophoblastic cells, and their carbohydrate receptors are present on the uterine epithelium, and these they mediate the attachment of the embryo to the uterine epithelium. So 2 layers are formed from the trophoblast. One is the cytotrophoblast, other is the syncytial trophoblast. The cytotrophoblast is cellular layer that is closest to the inner cell mass, that is embryoblast, and syncytial trophoblast is the cell wall breaks down on the side near the uterine wall. It is more invasive in nature. It is the layer which is also responsible for the secretion of human chorionic gonadotrophin hormone as well and maintenance of the pregnancy. And in syncytial trophoblast, the cells do not have any cell boundaries, and these cells are present like syncytium of the nuclei. By day 10, the blastocyst is completely embedded. So here you can see this is the blastocyst, this is uterine epithelium. Here you can see the glands as well. This is the functional zone of the endometrium, also called the stratum functionale. Here you can see reorientation of the blastocyst so that its embryonic pole where the embryoblast is present, it faces the uterine endometrium, and this pole is called embryonic pole. Here you can see the syncytiotrophoblast, which is more invasive in nature, and it has started to invade the uterine endometrium, and the process of implantation has started. More and more invasion by the syncytiotrophoblast. Also at this stage, lacunae starts to develop in the syncytiotrophoblast. This is the cytotrophoblast, and here you can see the amniotic cavity has started to begin. So another clinical correlate over here, very important. Normally the after the fertilization, implantation takes place in endometrium of the uterus. So any implantation other than the endometrium of the uterus is called ectopic implantation. So ectopic implantation can take place into the uterine tube, which is the most commonest type. Then it can implant in the cervix, into even the abdominal cavity, and sometimes the ovaries as well. So blastomeres, there are certain definitions you must understand. The blastomeres are non-differentiated cells and can give rise to any tissue. So the stem cells are set aside and will continue to divide while remaining undifferentiated. Tissue specific means that can give rise to only one tissue. The pluripotent means can give rise to multiple different cell types. So the blastocystic inner cell mass cells are actually the pluripotent cells. Totipotential means the cells can give rise to any of the cell type. So there is a difference between pluripotent and totipotential cells. Totipotential cells can form any of the cell type, while pluripotent can form multiple different cell types, but not all of the cell types. So totipotential cells are formed at the morula stage, and pluripotent cells are present at the blastocyst. These are the inner cell mass. So the clinical correlate with this lecture are the first are the embryonic stem cells. These are derived from the inner cell mass of the embryo, and because these cells are the pluripotent stem cells, so these cells can be used to treat multiple conditions like the Alzheimer's syndrome, the Parkinson's disease, and the spinal cord injuries, and certain types of anemia as well. So these cells are formed, these are obtained by the process of the reproductive cloning. So there are the two disadvantages with this process. First disadvantage is that these cells do not resemble genetically with their host, so there may be the rejection process. And second disadvantage is that these cells are taken from the viable embryo, so there are the objections on the ethical points as well. So to overcome these problems, next new approach is therapeutic cloning or the somatic nuclear transfer. In this approach, the cells from the skin are taken, and these cells are then fertilized with an enucleated oocyte, and this oocyte is then stimulated to divide and form the blastocyst. So cells from this blastocyst are then taken to treat different conditions, and the two advantages of embryonic stem cells are now overcome because these are not taken from a viable embryo and all the genetic material is taken from the host. Another technique is the adult stem cells. So adult stem cells are taken from the adult. These are multipotent stem cells, not the totipotential or the pluripotent stem cells, and these cells can be used to treat certain conditions like Parkinson's disease. But the disadvantages of this approach are slow rate of the division and the scarcity of the cells. So dear students, here I complete my 2 days class. The references are the Langman's Medical Embryology. In the next class, we will continue with the second week of development. Thank you. Thank you very much students for your attention for the 2 days class, and Allah Hafiz till the next class.